The nervous system is one of the more complex systems of the body. So we're going to start out by doing an overview first of nervous tissue before we get into the more specific parts of the nervous system. After completing this segment, you should be able to list the functions of the nervous system, list the cells found in the nervous system and tell the general function of each cell, describe the structure of a neuron and list the general function of each of its parts, list the structural and functional divisions of the nervous system, explain what happens during a nerve impulse transmission, and list the parts of a reflex arc and tell the function of each part. There are three general functions of the nervous system. It monitors the environment, integrates information, and then responds to the stimuli that it gets from the environment. Now, in order to monitor the environment, we have to have receptors. So one of the structures of the nervous system are the receptors who pick up signals that we refer to as stimuli. This is sent to a central location where the stimuli are analyzed and decisions are made about how to respond. Once the decision has been made, response signals are sent to various effectors. These are muscles or glands that respond to the stimuli in a way that helps us maintain homeostasis. For example, you see a glass of water and that sensory input is sent into your brain. Now, in your brain, you're doing some integration here. You're registering that you've seen water, you're registering that maybe your mouth feels a little dry, and you decide that it would be good to drink the water. Now you send that motor information out along some nerves to the muscles in your arm so that you pick up the glass of water and you drink it. The nervous system can be classified based on its structural composition. The central nervous system, or CNS, is composed of the brain and the spinal cord. The peripheral nervous system, or PNS, are the nerves that are attached to the brain, which are the cranial nerves, and the nerves which are attached to the spinal cord, or the spinal nerves. If we look at a functional classification of the nervous system, we only have to look at the peripheral nervous system structures. The peripheral nervous system is divided into the sensory division. This is when sensory information is conveyed to the central nervous system. Somatic sensory fibers pick up information from the exterior of the body. When we use the word somatic, think in terms of conscious. This is what you're aware of. There is also visceral sensory fibers that take sensory information from your internal organs. Typically, you are unaware of the sensation that you're feeling in your internal organs. The other functional classification is the motor division, or the efferent division. And the motor division takes impulses from the central nervous system to the various effectors. The motor division is further subdivided into the somatic nervous system, which is your voluntary nervous system. Here the skeletal muscles are the effectors. So again, these are the conscious movements that you make. The other division is the autonomic nervous system. This is involuntary. Here your smooth muscle, your cardiac muscle, and your glands are involved. And you can't consciously control any of these, and yet they need to be controlled to help you maintain homeostasis. The autonomic nervous system is further subdivided into the sympathetic and the parasympathetic nervous system. Now we'll look at all of these in a little more detail a little later on, but just to give you the roadmap here. Central nervous system is brain and spinal cord. The peripheral nervous system are all the nerves. Now when we get into the peripheral nervous system, you've got the sensory and you've got the motor division. Sensory can be somatic, your conscious sort of things, or visceral. But the sensory division carries information from either conscious parts, touch for example, or the unconscious parts, just the fact that your stomach is full or empty. And the motor division carries information from the central nervous system to the effectors. Now the somatic nervous system is your voluntary or conscious nervous system. So here the motor effectors are the skeletal muscle. The autonomic nervous system is the visceral nervous system. This is your unconscious part. So it's going to conduct impulses to the heart, to the organs of the body, and to the glands. The autonomic nervous system is further divided into your sympathetic division and your parasympathetic division. Your sympathetic division is your fight or flight division. The parasympathetic division is your more day-to-day housekeeping kind of system. Let's take a look at nervous tissue. There are two types of cells in the nervous system. One type are the supporting cells, or the neuroglia. The other type is the neurons. Now, the neuroglia support, protect, and insulate the neurons. The neurons are the cells that conduct nerve impulses. When we think of nerves, it's the neurons that we're thinking of. These cells are excitable and conductive. There are 6 kinds of supporting cells, or glial cells. In the central nervous system, the neuroglial cells are the astrocytes, which help produce or form the blood-brain barrier, the microglia, which are phagocytic cells, the ependymal cells, which are involved in cerebrospinal fluid formation, and the oligodendrocytes, which form the myelin sheath on the nerves inside the central nervous system. Gliomas are tumors of these supporting cells. The glial cells are the only cells of the nervous system that can still divide. Neurons lose the ability to divide in young adulthood, but since these cells can still divide, these are the ones that are likely to undergo changes in cell division, and you'll remember that's how cancers are produced. So brain tumors typically involve one of these cell types, and they're generally called gliomas. The blood-brain barrier here, produced by the astrocyte, involves the astrocyte putting a whole extra layer around the capillaries. A number of things that are found in the bloodstream, if they were allowed to get into the brain tissue, would damage the brain tissue. So the astrocytes simply put an extra barrier between the capillaries and the brain tissue, slowing down the diffusion of certain materials from the blood to the brain cells. The microglial cells are phagocytes. They can kind of wander around among the neurons, eating up little bits of cellular debris, bacteria, things like that that may get into that system. The ependymal cells line the hollow spaces in the brain, the ventricles, and again, cerebrospinal fluid is produced from the blood, and these cells simply act as an extra filtering unit to minimize some of the material that gets from the blood into the cerebrospinal fluid. And the oligodendrocytes send out extensions of their cell membrane to wrap around the nerve cells to make the myelin sheath. The myelin sheath is like the insulation on wires. It keeps nerves from short-circuiting with each other. In the peripheral nervous system, we have 2 glial cells. One is the Schwann cells, which make the myelin sheath in the peripheral nervous system, and the other one are the satellite cells, which help cushion and protect. And here you see some satellite cells around part of a neuron. And these little marshmallow puff things are the Schwann cells, making up the myelin sheath. The neurons are irritable and conductive cells, and this is important because they have to transmit nerve impulses, and that is their function. And there are 3 parts to a neuron. There are the dendrites, the cell body, and the axon. Neurons have numerous dendrites. They're short, highly branched structures. These are the parts of the neuron that are receptive to information. They're going to be attached or close to receptors and be able to pick up information there. The cell body is the large part of the neuron. Most of the cytoplasm is here in the cell body. The nucleus is in the cell body, and most of the organelles are in the cell body. The metabolic processes of the neuron, for the most part, occur in the cell body. Then the axon is a single long projection off of the neuron, may go feet. As a matter of fact, it can be quite long. This is the part that's going to be myelinated. That's what these little marshmallow puff things are looking like here. And this is what's going to transmit the nerve impulse to the next cell, either the muscle cell or another neuron or a gland cell. Now, when the axon gets to the end, it branches into these axon terminals. And this is the part of the cell, of the neuron, that's going to communicate with the next cell in line. The axon terminals are at the ends of the axon, and they're going to release neurotransmitter. While we're going to use an electrical kind of impulse along the neuron, when we get to the end, we're going to have to use a chemical in order to transmit the impulse to the next cell. The myelin sheath, sometimes referred to as the neurolemma, is made of numerous Schwann cells that wrap themselves around the axon. Myelin sheaths in both the peripheral and the central nervous system are insulators. Just like you need insulation on wires when they're grouped together, you're going to have a bunch of axons making up a nerve, and so you want them all insulated. The insulation also causes faster nerve transmission to occur. Now, in the peripheral nervous system, there's going to be space between the Schwann cells, and these little spaces are called the nodes of Ranvier. So if we look back at our neuron, individual Schwann cells wrap, and then there are little spaces between, and these are the nodes of Ranvier. Multiple sclerosis is an autoimmune disease where the body starts to destroy the myelin sheath of the peripheral nerves. As a result, the nerves begin to short circuit, and because the myelin sheath helps nerve transmission to be faster, nerve transmission slows down. Now, this disease shows itself by a progressive loss of muscle function, not because the muscles are incapable of functioning, but because the muscles fail to get the appropriate signals to function. And just what you need, some more terms to add to your ever-expanding anatomy and physiology vocabulary. In the central nervous system, if we have a cluster of cell bodies, we're going to call that nuclei. And if we have a cluster of cell bodies in the peripheral nervous system, we're going to refer that as a ganglion. Same thing, but in different locations, they're going to have different names. In the central nervous system, if you have a bundle of nerve fibers, and these will primarily be axons, we're going to refer to that as a tract. But in the peripheral nervous system, that same bundle of nerve fibers is referred to as a nerve. White matter are simply myelinated axons. The myelin sheath is full of fat since it's basically cell membrane, and fat is like lard, it's white. Gray matter will be any of the unmyelinated fibers in cell bodies. So the dendrites, the cell bodies, and a few axons that are not myelinated will compose the gray matter. Nerves can be classified based on how they function. Sensory or afferent nerves have their cell bodies outside the central nervous system. Their dendrites are associated with receptors, which are going to be close to the surface of the body or close to organs in the body. The receptors will collect information and will transmit it through the axon to the central nervous system. So these are input neurons. Now, receptors can be very simple, like touch receptors, some of which are basically just bare dendrite endings, to very complex, like the eye and the ear, which have a number of structures that play a role in collecting the data and transmitting it to the sensory neuron. Another group of neurons are the motor or efferent neurons. Now, these carry impulses to an effector, either a muscle or a gland. Their cell bodies are inside the central nervous system. Association or interneurons are connectors. They help connect sensory neurons to motor neurons. They're part of that integration system. Their cell body is inside the central nervous system. As a matter of fact, the entire neuron is inside the central nervous system. As we've said before, neurons are irritable cells that are capable of conducting nerve impulses. So let's look at the physiology of that nerve impulse. Excitable cells, like nerve cells and muscle cells, have something called a resting membrane potential. That is, we say they are polarized. What this means is that if the yellow line here is the cell membrane, it's a little more positive outside the cell and a little more negative inside the cell. So there's a little bit of polarity. It's not exactly equal. Now I know we've said Mother Nature just loves equality, and equality is still here. There's the same number of positive ions outside the cell as inside the cell. There's the same number of negative ions outside the cell as inside the cell. However, the positive ions outside the cell are sodium, the positive ions inside the cell are potassium. This appearance that there's more positives outside the cell and more negatives inside the cell is not based on actual numbers of ions, but simply on how they're arranged. Mother Nature does a funny little thing with the cell membrane where she's kind of pumping positives out and they tend to stay clustered here, and negatives then try to come up and follow the positives out, so they sort of cluster on the inside. So if we're going to generate a nerve impulse, this is a little piece of axon right here with its resting membrane potential going on, its polarized state. Slightly positive on the outside, slightly negative on the inside. Remember, this is based on an arrangement of ions, not on a number. You'll also remember that the membrane is impermeable to ions, which means the only way ions can get across is if there's some sort of protein channel for them. And normally the ion channels are closed so that we don't have free flow of ions back and forth across the cell membrane. In order for an action potential to develop, we need to depolarize the membrane. And what happens is a stimulus opens one of those sodium gates. Now, it only opens one of them, right there where the stimulus is. Because there's more sodium outside the cell than inside, if you open a door for it, the sodium rushes in, bringing with it its positiveness and making it a little more negative out here. So we kind of reverse the polarity of the membrane in that one spot. That's the beginning of an action potential. Now, because we change the polarity in this spot, this triggers the opening of the sodium gate next door, and we change the polarity in that spot, which opens the sodium gate next door, and we change the polarity of that spot. So before you know it, we've got this chain event, this domino thing occurring where we change one, which changes the next one, which changes the next one. This is what we call the propagation of a nerve impulse, the fact that once we get it started, it continues to go, just like flipping one domino and they all fall over, but one at a time. Now, in order for the nerve to function again, we've got to get it back to its resting membrane state, and that's called repolarization. Now, this same change that opens sodium gates, allowing sodium to rush in, those gates are only open momentarily and then they naturally close again. But as they're closing, potassium gates open. Now, potassium is more prominent inside the cell than outside, so when we open a doorway for potassium, it's going to rush out, taking its positiveness with it. Usually we overdo this a little bit because opening and closing the gates is not a very specific thing, and we tend to make it even more positive outside the cell than it was to start with. We call this hyperpolarization. But the sodium and potassium pumps kick in and completely restore the balance, making just the right number of sodium ions on the outside, just the right number of potassium ions on the inside, and we go back to being polarized. That resting membrane potential is returned. Now, a nerve cannot conduct an impulse again until we have repolarized it. So all these events we've just talked about take place faster than you can possibly imagine. Now, the action potential will go on until we run out of neuron. And nerve cells terminate in those axon terminals. Nerve cells do not directly attach to the cell with which they're going to communicate, whether it's another nerve cell or a muscle cell or a gland cell. There's a little space between the nerve cell and the next cell, and that's called the synapse. The little space is the synaptic cleft. And in order to move or stimulate this next cell, the axon releases a chemical called a neurotransmitter. And this chemical can diffuse across the synapse, attach to receptors for that chemical on the other cell, and that can cause sodium gates to open, and we get an action potential going on that cell. Now, different things can cause depolarization. Some receptors are susceptible to light, some to mechanical changes, vibration, pressure, some to electrical, and some to chemical. And those that are susceptible to chemical, this is where the neurotransmitters can cause that depolarization event to occur. All of these things can cause sodium gates to open on the neuron to initiate the action potential. Once depolarization starts, we say it's self-perpetuating. One sodium gate opening causes the next sodium gate to open, causing the next sodium gate to open. And of course, opening sodium gates, they're going to close immediately, and potassium gates will open as a part of that whole event. Nerve impulses require a lot of energy because you've got to fuel those ion pumps. The sodium and potassium pumps take ATP in order to restore resting membrane potential. As a result, nerves need a good supply of oxygen and nutrients to generate that ATP. If you apply cold to an area or pressure to an area, you inhibit blood flow. And before long, your nerves can't keep generating ATP. They can't return the nerve back to resting membrane potential. And if it can't go back to resting membrane potential, we can't generate another nerve impulse. So those areas will start to feel numb or get that go-to-sleep feeling. As soon as you warm it up or relieve the pressure and the blood flow comes back again, you can again get resting membrane potential on the neurons, and feeling comes back into the system, and you can generate impulses again. Sedatives and anesthetics work primarily by inhibiting the opening of sodium gates. So even though there may be nerve impulses bombarding the membrane of the neuron, if we can't get the sodium gates open, we can't start that action potential. No action potential, no nerve transmission. Reflexes are a set of rapid, predictable, and involuntary movements that are done in response to certain stimuli that help you maintain homeostasis. Basically, they're protective. They try to pull you away from potentially dangerous situations, help you maintain balance, things like that. And your reflexes can be divided into the autonomic reflexes. This would be if smooth muscle or cardiac muscle is involved in the reflex, or a gland is involved in the reflex. And somatic reflexes, which are when skeletal muscle is involved. Now, reflexes are based on a reflex arc. First of all, you have to have a receptor for the stimulus. A sensory neuron has to then take the information to an integration center. Now, this integration center will work in two phases. In one phase, it's just an automatic, this is what we're going to do. In the other phase, there is a little more contemplation about analyzing the input. But initially, the integration system has already built in, it's already hardwired in. If this happens, you're going to do this. We'll ask questions later. So there is a motor neuron that will take certain information to an effector. Now, if it is a somatic reflex, the effector will be a skeletal muscle. If it is an autonomic reflex, the effector will be smooth muscle, a gland, or cardiac muscle. So let's think about this a minute. Have you ever had someone jump out in front of you and say boo? That would be your stimulus. Your integration center initially said, I'm frightened, I'm going to jump, I'm going to respond, and you had that response no matter what, even though nanoseconds later you recognized it as someone you knew and someone who was not threatening to you. And that's when you felt really foolish and you laughed. So you had that automatic, I'm going to respond this way no matter what, and then you thought about it just for a little bit and you realized that there was no threat. There was no reason for you to respond this way. This is how a reflex works. That is, some information, this happens, you're going to do this, we're not going to think about it. And then when you do think about it, you may change or modify your behavior a little bit because you overreacted. Now, a lot of your reflexes are spinal reflexes because the integration center is in the spinal cord. You don't really think about it. For example, a pinprick is unpleasant. That is sent in along this sensory neuron. There is an association neuron, an interneuron here, that connects across to a motor neuron. Because that was unpleasant, you jerk back from it. That's called a withdrawal reflex. And that would be a somatic reflex because a skeletal muscle was involved. Some examples of somatic reflexes. One is your patellar reflex or your knee jerk reflex. If you hit the patellar tendon just right, it makes your body think that your quadriceps are being overstretched, and so they contract quickly, and that kicks out your foot. The other one is the one we just talked about, the withdrawal reflex. You encounter something unpleasant, a pinprick or something that feels slimy or gooey, and you pull back from it before you really assess what it was that is doing that, making you do that. Autonomic reflexes are going to involve the heart or glands or smooth muscle. For example, if you get hot, you sweat. You can't control that. You just sweat. You get embarrassed, you sweat. You would like to control that, but you can't. Another thing is when you're frightened, your heart rate goes up. You can't stop that from happening. It's a reflex. It's an autonomic reflex because the cardiac muscle is involved. Now, physicians frequently check reflexes when they're assessing nervous system function, because if you have abnormal reflexes or super reflexes, distorted reflexes, that can indicate certain nervous system disorders. Let's do a quick check here. The part of the neuron that transmits a signal to the next cell would be the axon, the dendrite, the cell body, or the nodes of Ranvier. It would be the axon. The ion that enters a cell to induce an action potential is potassium, calcium, sodium, or acetylcholine. Sodium enters the cell. Potassium exits the cell. Now, acetylcholine we didn't mention earlier, but it's one of the neurotransmitters that can be released by the axon terminal. The glial cell that is responsible for phagocytosis is the oligodendrocyte, astrocyte, microglia, or ependymal cell. The microglia are the phagocytic cells. The oligodendrocyte is in the central nervous system making the myelin sheath. The astrocyte helps with the blood-brain barrier, and the ependymal cell helps with the production of cerebrospinal fluid. A reflex is a rapid, involuntary, and predictable response, is usually mediated by the spinal cord, helps maintain homeostasis, or all of the above. The correct answer this time is all of the above. Most of your reflexes do go through the spinal cord. There are a few that go through the brain, but not very many. They are rapid, predictable, and involuntary, and their function is to help you maintain homeostasis.